Hi, my name is Mohamed. I'm here with the Summer Institute on Medical Ignorance. Here in Dr. Louise Hecker's lab, we use a special plate reader. This is the $23,000 Promega GloMax multi-detection system. At that price, you might be asking, what does it do? Well, it measures absorbance, luminescence, and fluorescence, which is just a fancy way of saying light emission or absorbance of compounds. Now, why is it so important? This is important in research since companies have created assay kits which help quantify systems in biology and turn your study into tangible data. An assay is just a procedure performed which helps measure the presence of a target in a culture plate. But what am I using it for? Currently, I'm using an assay kit called Amplex Red Hydrogen Peroxide Peroxidase Assay Kit, which helps me detect the presence of hydrogen peroxide in cells, which is a stress signal. Here's how it works.
Here is the structure of Amplex Red, and this is HRP, a peroxidase which recognizes hydrogen peroxide, binding to it and driving the reaction from Amplex Red to resirufin, producing O2 as a byproduct. The plate reader I mentioned excites resirufin with light at a specific wavelength. Resirufin will then emit a light at different wavelength, producing a fluorescence signal the plate reader can record.
Here's how the machine works. First, you must turn it on. And then you must select the protocol appropriate to your assay and the size wheels. And then you have to insert the fluorescence kit if you're using fluorescence. Now that the fluorescence filter is in, I can put the plate. I'll be using a 96 well black clear bottom plate. Now that that's in, and I can start. Now we can export to Excel. Now we can make an average of the data. And now we can subtract out the blank. Why do we do that? Well, because there's background noise. Otherwise, the data is just arbitrary. Now that we have meaningful numbers, we can do standard deviation. And now I can put it on a bar graph. Let me add the standard deviations, error bars. We do a plus with a cap, then let me specify. This allows us to see that with increasing concentration of hydrogen peroxide, we have increasing fluorescence. This plate reader, and specifically this assay kit, helps my lab collect meaningful data. Well, what do we use this data for? We can use it to test certain compounds and measure the oxidative stress signaling, which is the hydrogen peroxide we've been testing, and see how much of it is produced if we treat it with this drug, and maybe if we can decrease it if we treat it with a different drug. This would be a great way to produce a new drug in the market which will help people worldwide. Since in my lab we use HEC-NOX4 cells and HPA-EC cells, and this assay seems to work fine. My question is, does it work on all cells?